Romanovs had different teams of scientists beefing it out for half a decade, claiming the other teams were wrong and conducting their own DNA tests. The results and conclusions from the first two Romanov DNA papers, the identification of the remains from the grave near Yekatarinberg, and the comparison with the DNA of Franziska Szankowska, who had claimed to be Grand Duchess Anastasia, were not enough to convince everyone. Two Romanov children were still missing, but the second half of the 1990s, when the field of ancient DNA studies was still very new, saw a flurry of more Romanov DNA papers and one mysteriously unpublished paper. These tests focused on Tsar Nicholas and his wife, Tsarina Alexandra, and they sought to answer the same question. Had the last emperor and empress of Russia really been found? The skeletal remains of 4 adult men were excavated from the grave in the forest near Yekaterinburg. 3 male servants have been executed alongside Tsar Nicholas and his son, 13-year-old Tsarevich Alexei, the physician Dr. Eugene Botkin, the valet Alexei Trupp, and the cook Ivan Kharitonov. The identification of one of these skeletons, labeled as skeleton 4, as Tsar Nicholas, rests on 2 types of DNA. Short tandem repeats found in nuclear DNA, DNA in your cell's nuclei that you inherit from both of your biological parents, and mitochondrial DNA, the DNA in your cell's mitochondria that you inherit only from your biological mother. Skeleton 4's short tandem repeats matched 3 of the grave's female skeletons in patterns that we would expect if skeleton 4 was their biological father. This fits with the Romanovs if those 3 skeletons belong to 3 of Nicholas's 4 daughters. Skeleton 4's mitochondrial DNA sequence matched 2 living maternal relatives of Tsar Nicholas, except for one thing: a mutation called point heteroplasmy. Heteroplasmy is when an individual has 2 or more distinct mitochondrial DNA sequences in their cells. Point heteroplasmy is when this happens at a specific point, a base, also called a nucleotide, in the sequence. When we talk about a DNA sequence, what we mean is the bases, the nucleotides adenine, guanine, thymine, and cytosine, symbolized by the letters A, G, T, and C, lined up together in a specific order. So at each position in the sequence, you'll find one of these 4 nucleotides, but not in the mitochondrial DNA sequence of skeleton 4, identified as Tsar Nicholas. At position 16169, Nicholas had a mixture of the 2 bases cytosine and thymine, C and T. The 2 living maternal relatives to whom his DNA was compared did not have this. At this time, heteroplasmy was thought to be really rare. In order to verify that Nicholas had this point heteroplasmy, and that skeleton 4 really is Nicholas, we need to look at DNA from someone more closely related to him. Grand Duke Georgij Alexandrovich Romanov didn't live to see the Russian Revolution and his family's fall from power. Born in 1871, he was Nicholas's younger brother, and when Nicholas became emperor, George was actually the heir to the throne because Nicholas hadn't had children yet. However, George was diagnosed with tuberculosis, and there wasn't a treatment for this disease at the time. Honestly, it can still be hard to treat tuberculosis now. At his doctor's suggestion, George went to live in the spa town of Abastuman, which is now Abastumani in Georgia, in the hopes that the dry mountain climate would help improve his health. It didn't, and he died there in 1899. George was buried in the Saints Peter and Paul Cathedral in St. Petersburg. This means we have someone closely related to Nicholas, who would share his mitochondrial DNA sequence with a known burial location. Scientists took samples from George's femur and tibia, and the article published about this actually has photos of the bones after the samples were taken. The bone samples they took for DNA extractions are huge, and I wanted to bring this up as a way of just briefly reflecting on how far the field of ancient DNA has come over the past 30 years. When we drill bones for DNA samples now, we don't take that much, and we tend to drill from parts of the skeleton that won't be as noticeable as a giant chunk missing from a femur. We're often drilling teeth, though in the case of George and Nicholas, samples had been taken from the femur and tibia of skeleton 4, Nicholas, so this gave the scientists a consistent sampling strategy. These tests included another comparison with the mitochondrial DNA sequences of 2 living relatives of Nicholas and George. These were Countess Xenia, a descendant of their sister, who was also named Xenia, and James George Alexander Bannerman Carnegie, the 3rd Duke of Fife and Chief of Clan Carnegie. He was a descendant of Nicholas's aunt, Queen Alexandra of Great Britain, who was the sister of their mother, Tsarina Maria Feodorovna. Now that we're starting to get some more characters in this part of the story, let's start adding them to our family tree here. So here's George. Nicholas and George had a couple sisters, one of whom was Xenia. And if we go down her family tree of female descendants, we then get Countess Xenia. For our other living maternal relative at the time of this study, we need to go back up to Nicholas and George's grandmother, Louisa of Hesse-Kassel, Queen of Denmark. So one of her other daughters was another Alexandra, the daughter-in-law of Queen Victoria and thus later Queen of Britain herself. So through her female line descendants, we get the Duke of Fife. Okay, so now this is our completed mitochondrial DNA family tree for Tsar Nicholas. Their mitochondrial DNA sequence matched the sequences from the individual identified as Tsar Nicholas, except for the point heteroplasmy. Neither Countess Xenia nor the Duke of Fife had this mutation. So what about George? George's mitochondrial DNA sequence matched the sequences of Countess Xenia and the Duke of Fife, except for the point heteroplasmy. At position 16169, George had the same mixture of the nucleotides cytosine and thymine observed in skeleton 4, the individual identified as Tsar Nicholas. Both brothers having the same mutation in their mitochondrial DNA at the same position in their sequences makes it likely that the mutation occurred in their mother, Maria Feodorovna, and then she passed it to them. But we can't really know that with more certainty without a DNA sample from her too. Further studies also showed that mitochondrial DNA heteroplasmy is more common than previously thought. You would think that these results would make for a much more convincing positive identification of the remains of Tsar Nicholas, but no, apparently not. More controversy came from samples of biological material collected from Nicholas before his death. In 1891, before Nicholas became Emperor of Russia, he was visiting Japan when he was attacked by a random dude wielding a sword. The bloodstained handkerchief that had been used to treat the resulting head wound ended up at a Japanese museum. In 1997, a team of scientists claimed to have successfully extracted and analyzed DNA from the bloodstained handkerchief that didn't match the DNA profile previously obtained for Nicholas. But they didn't publish their study or go through any peer review process. We've seen this before with some of the DNA studies of Caspar Hauser that also use bloodstained fabric as their source of DNA. Without a published article, we can't know what extraction protocols they used or what kind of decontamination procedures their lab had in place. We can't know their methods, what results they obtained, or how they reach their conclusions. The peer review and publication process is how dialogue takes place within scientific communities. It's part of how we learn what methods work and what doesn't work, and why. This has a huge role in how we design new projects. It really is a big part of how we communicate with each other, and it's not unusual to be asked, hey, do you have an article or citation for different methods and workflows for analyses or tools? That's why it's super weird and frustrating to learn that a study was done in response to an initial study, that the results challenged the results and conclusions of that initial study, but it wasn't published. Things get even weirder. That same lead author of the not published bloodstained handkerchief study then led another project trying to extract mitochondrial DNA from 25 hairs attributed to George. They claimed the sequences obtained from these hairs didn't match the mitochondrial DNA sequences obtained from Nicholas, and that it lacked the point heteroplasmy observed in the DNA that came out of George's bones. These results could be the result of contamination. They might have been looking at the DNA of someone who wasn't George, but it's difficult to know. They did publish this paper, but they published it in a Japanese journal, so it's difficult to access and there doesn't appear to be an English translation. I wasn't able to get access to the original study, so the reference I have for it is in a different paper that cites it. Another team of scientists then launched another study and published a paper that starts with claiming that the samples used in the original Romanov DNA study were contaminated. Their big problem with the original study was the size of DNA fragments obtained from the 9 individuals buried in the grave near Yekatarinberg, so it's time to beef it out over base pairs. We've already talked about the 4 nucleotides that create DNA: adenine, thymine, guanine, and cytosine, which we also refer to as bases. These create the 2-stranded double helix structure we all know and love by pairing up with each other. Adenine pairs with thymine, and guanine pairs up with cytosine. When we talk about the length of a DNA sequence or the size of a gene, we measure it in terms of how many base pairs long it is. One of the things that makes ancient DNA so difficult to work with is the fact that DNA degrades over time. The sequence falls apart, the bonds that hold nucleotides together break, and we're left with fragments, which again, we measure in the number of base pairs. With ancient DNA, we can expect fragments that are maybe a few hundred base pairs long, but we need to remember that factors such as the environment or how samples are handled and stored can influence how well ancient DNA is preserved. I'm saying that now because we're going to come back to it later. In the original Romanov DNA paper, they obtained DNA fragments up to 1,223 base pairs long. This new team of scientists, Knight et al., claimed that those fragments were way too big to be ancient DNA and challenged those results and conclusions with their own study. For the subject of their study, Knight et al. Chose Tsarina Alexandra's older sister, Grand Duchess Elizabeth Feodorovna. Born in 1864 and nicknamed Ella, she married Grand Duke Sergei Alexandrovich Romanov, the brother of Tsar Alexander III and uncle of Tsar Nicholas II. After Sergei's assassination in 1905, Ella founded a convent, the Order of the Saints Martha and Mary. She was arrested by the Bolsheviks in April 1918 and was executed with other members of the extended Romanov family in July, shortly after the execution in Yekatarinberg. Ella and her companions had been thrown into a mine shaft during their execution, and their bodies remained there until they were exhumed in October. The bodies hadn't decomposed too much, so they were still recognizable enough to be identified, and they were autopsied. Ella was initially buried in a cathedral near the execution site, but her remains were moved to the St. Seraphim of Sarov Church in Peking in 1920, and then moved again to the Russian Orthodox Church of St. Mary Magdalene on the Mount of Olives in Jerusalem. She was canonized by the Russian Orthodox Church outside Russia in 1981, and canonized again by the Moscow Patriarchate in 1982. There's a figure of Ella carved over the west door of Westminster Abbey. In 1981, Ella's coffin was opened, and a bishop of the Russian Orthodox Church abroad removed one of Ella's fingers to keep as a relic in a reliquary. This relic was then taken to New York, and that's where samples were obtained for DNA testing. The DNA samples were extracted and analyzed at two independent labs, a lab at Stanford University and Los Alamos National Laboratory. Yes, this is the same place where the Manhattan Project developed the atomic bomb. They weren't able to obtain DNA fragments longer than 250 base pairs. The sequences of mitochondrial DNA they obtained from Ella didn't match Alexander's sequence. Remember, they're supposed to have the same mitochondrial DNA sequence they inherited from their mother, Princess Alice of Great Britain. Thus, they argue that the Romanovs may have been misidentified. So, what's going on here? How can we make sense of this? Thankfully, an article published later critiqued this study, as well as the two studies challenging the identification of Tsar Nicholas, the studies with the blood-stained handkerchief and the hairs attributed to his brother, Grand Duke George. So, what did this article have to say about all of this back and forth contamination and base pair drama? First, this article explained that a mixture of mitochondrial DNA sequences had been found in the Ella study. That sounds like a contamination red flag, and the sequences they obtained also didn't match Prince Philip, Duke of Edinburgh, the maternal relative to whom Alexander's DNA was compared. If we think back to the post-mortem treatment of Ella's remains and compare it to this grave excavated near Yekatarinberg, there were more opportunities for contamination. First, Ella's body was removed from the mineshaft where she had died, identified, and autopsied before being placed in a coffin. Then her remains were moved multiple times to different environments, and these different environments will have had a role in the preservation of Ella's DNA. Different environmental and storage conditions, including climate, temperature, and soil conditions, can preserve DNA differently, influencing how fast it degrades over time. Cold environments preserve DNA better, and it's similar to how certain environments can preserve soft tissue and create natural mummies. The grave near Yekatarinberg isn't in Siberia, but it's right next to Siberia, and the individuals stayed in that grave for decades. Their DNA might not have degraded as fast as Ella's DNA, resulting in those longer DNA fragments. Ella's remains encountered additional opportunities for contamination when her coffin was opened for the removal of some of her remains as relics, which were then again moved to a different location. There are also some odd characteristics in the overall research design of the studies of DNA from Ella's finger, Nicholas's bloodstained handkerchief, and George's hair. They didn't evaluate their data using the same framework as the original Romanov DNA study or the study of George's bones. The original studies tested two opposing hypotheses. The remains from Yekatarinberg are the Romanovs, or they are not the Romanovs. Part of this involved a lot of statistics, but they also included verified positive control samples from living relatives of the Romanovs. The finger, handkerchief, and hair studies didn't do this. They didn't introduce new samples from other living relatives, and if the authors of the Ella study are arguing that the samples from Yekatarinberg are contaminated, then that implies they must be contaminated by an individual who just happened to share the same maternal lineage as Prince Philip, who has the same maternal lineage as Tsarina Alexandra. Yeah, it's a bit of a stretch. The finger, handkerchief, and hair studies didn't really overturn the results of the original studies. However, there is still lingering controversy. 2 Romanov children, Tsarevich Alexei and one of his sisters, are still missing at this point in the story. The discovery of a second grave near Yekaterinburg and the DNA analysis of its occupants will be discussed in part 4 of this series. But those studies are not the end of the Romanov DNA story. A couple of very recent studies will bring everything full circle, so I hope you'll join me for the next chapter. Thank you for watching.